Good evening, everybody. Thank you for joining us. I'm delighted to welcome you all to our ASSFN webinar, where we're going to hear from established and emerging functional neurosurgery leaders in the field of brain-computer interfaces, which is really a very exciting and rapidly growing field. Before we get started, I wanted to let everybody know that they can learn more about the ASSFN and also have access to these webinars, which are recorded both on our YouTube channel, but also through our website, ASSFN.org. And also, we encourage you all who are not members to become members of the ASSFN, and you can do that also directly through our website. I also wanted to let you all know that our biennial meeting that happens every other year will be happening this year. It'll take place in Cleveland on May 30th through June 2nd. There will be a lot of exciting functional neurosurgery content and certainly a lot of content surrounding this particular topic as well. So we encourage you all to attend. To get us started, I'm delighted to introduce David Brandman, who's an associate professor and functional neurosurgeon at UC Davis, who is himself an emerging leader in this field, and who will be moderating this session. Thanks, David.
Outstanding. Sharona, thank you so much for that kind introduction. I'll go ahead and share my screens. Hopefully you can see the main presentation, not the presenter view. Yeah, so I'm David Brandman. I'm an associate professor at University of California, Davis. I've been on staff here for just over 4 years now. I'm moderating today's session, and I'm really excited to be here today. By way of introductions, just starting quickly with my disclosures, I'm currently a consultant for Globus Medical, and I'm the proposed study PI for an upcoming early feasibility study together with Paradromics Incorporated. I'll talk more about that in just a moment. So now is the time to have this conversation. It's a very exciting time to be talking about brain-computer interfaces. I wanted to start by telling you what a brain-computer interface is for the purpose of today's presentations. A BCI is a device that records brain signals from people that are paralyzed and then translates those brain signals to allow people to control objects in their environment. So fundamentally, a BCI has 3 parts. There is a source of brain signals. There's what you're trying to control, or an effector, and then the map that connects the two. So it's helpful to think about this kind of technology as having 3 parts: a sensor, a decoder, and an effector. And by and large, when we talk about BCI sensor technology, most research that happens internationally uses electroencephalography or other kinds of non-implantable approaches. But given that the focus here today is about functional neurosurgery, we wanted to focus on implantable brain-computer interfaces and the kinds of possibilities that these technologies offer patients living with paralysis. And as we're about to hear later in these presentations, the field for BCI is several decades old, and we've been entering the world of clinical trials for about 20 years now. But we're at a very exciting point where industry is now coming in to lead early feasibility studies, studying the safety profile of implanted wireless devices that hope to restore communication. And so as an example of this, some of the sponsors on this some of the industry members in this clinical trials have completed early feasibility studies or currently plan to launch early feasibility studies. In my own role as proposed study PI for Paradromics, we hope to be launching an early feasibility study later this quarter, if not early next quarter, looking at fully implanted devices to restore communication for people with paralysis. So with that in mind, I wanted to go ahead and introduce today's speaker, starting with Matt Wilsey. So Matt's a friend and an assistant professor of neurosurgery at University of Michigan Health. His own research is focused on chronically implanted Utah rays as part of the BrainGate clinical trial, showing how this kinds of technology can be used to restore communication and independence for people with paralysis. I'm hoping that today he can speak a little bit about where we've been with BCI technology, some of the landmark demonstrations that have happened in both animal models and people, and kind of conclude in terms of the state of the art and where things are right now and hopefully where things may be going. Ben Rappaport is an assistant professor at the Mount Sinai and a practicing neurosurgeon there. So he's actually the co-founder and chief scientific officer of Precision Neurotechnology, leading the scientific development of ECOG-based BCIs. He's very active in the implantable BCI collaborative community, which I hope he's going to be speaking about later on today, as we think about how to get multiple stakeholders, the FDA, payers, industry, clinicians, scientists together to make sure that we're all truly aligned moving this technology forward and bringing it to the patients that need it most. And then finally, Eddie Chang is currently chair of Department of Neurological Surgery, UCSF. Of course, he's had many contributions to the field through his career, but germane to this presentation today on BCIs, I've asked him to speak about his work in speech restoration and recordings of people in the epilepsy monitoring unit. So with that, I will go ahead and stop sharing. I think we'll take questions at the end today. So we'll start with Matt.
Thank you for the introduction. Hopefully you guys can see, I'm trying to go into slide view. Do you guys see my slides here?
Yes, we do.
Yes.
All right, perfect. All right, so thank you for giving me the opportunity to talk with you. It's great to be part of the field at this point in time. I'm going to kind of jump right into it here. So my disclosures. So I do have a research agreement with Paradromics, and we'll talk about a little bit about that at the end. Kind of what David had mentioned that I'll be talking about a lot of the history and motivation for brain-computer interfaces. I'll highlight some of the early BCI feasibility studies in animals and humans. We'll also talk about some of the BCI advances. We'll talk about kind of key BCI applications, future work, and a little bit about some of the clinical trials. So it probably is not appropriate to have a neurosurgery webinar without mentioning Penfield. Some of his early studies, intraoperative studies with electrical stimulation, really mapped out motor and sensory cortices, and a lot of the work that we've done has built upon some of his prior work. Some of the early neuroscientists, like in the early 1970s, 1960s, realized that animals could actually control individual single units in precentral gyrus, which implies that if you can control single units, something recording from single units could be controlled as well. And then some of the very modern BCI era kind of launched from some of the work done by Georgopoulos, which showed that during 2D arm reaches in animals, the spiking rate varied. So if you could record and measure the spiking rate, you could infer the direction of reaches. And a lot of our BCI work is, and these are just a couple of highlights. There's a lot of things that I haven't mentioned, but a lot of our work builds on some of these initial studies. I really don't have to motivate this brain-computer interfaces in this crowd. Obviously, paralysis, for example, is a really big problem. There's a lot of people with paralysis, and for some people that are paralyzed, it severely impacts their life. One potential therapy for these people are brain-computer interfaces. I'm showing a schematic from some of the work that we've done, showing a type of system that we typically use. A brain-computer interface is just that. First, you have to extract the signal from the brain. There's lots of places that you can't extract a signal. We have two, leading experts in ECOG with Dr. Rappaport and Dr. Chang, and so I'll let them talk about these devices in their studies. But a lot of the work that I've done have been with the use of microelectrode arrays. So a lot of individual arrays that are taken together and inserted at a predetermined depth into cortex. And this is an artistic rendition, but it shows the neurons surround the electrodes, the neurons fire, the electrodes record the signal. And based on what someone's trying to do, algorithms can interpret this and use it to control devices. Why the choice for microelectrode arrays? Well, these were designed so that they could be really close to the neurons, generate a high signal to noise, and have a lot of distinct information per channel. So this is one of the first feasibility demonstrations in animals. You're looking at a video of a non-human primate controlling a yellow computer cursor based on data that's trained from arm reaches, and the animal's trying to place the yellow cursor on the green dot, and presumably when it does this, it gets a reward. And so this is one of the first studies demonstrating cursor control with a brain-computer interface, and you can see that. And some of the videos we'll show later, the control has gotten much better than this. But shortly after this, in roughly 2002, a human study showed that humans could also be implanted with intracortical arrays in precentral gyrus and motor cortex. And when people attempted to make a movement, we can interpret the signals and use it to control computer cursors. So again, very early work, but demonstrates the feasibility of this technology. I'm going to highlight some of the other early applications for brain-computer interfaces. The picture on the left is one of the first demonstrations of a brain-controlled, one of the first of a brain-controlled robotic arm. This is a participant in the BrainGate clinical trial who's drinking from a cup. And then on the right is from the group at Pittsburgh. This is a robotic arm where the person has paralysis. They have electrode arrays implanted, and when they attempt to move their arm, the signal is interpreted and used to control robotic arms to manipulate objects and place it on a shelf, as you can see here. The important thing about these videos is these are real-time videos showing like these movements are being able to be manipulated and adjusted in real time. Here's a demonstration of a person now no longer controlling a robotic arm, but actually reanimation of the native limb. So this is instead of stimulating, instead of recording and then controlling a cursor, we're actually this is a group that Case Western actually stimulating the muscles themselves through functional electrical stimulation, and the person's actually able to reach out, grasp, and bring an object close to their mouth and take a drink. All right, so, since some of these early studies, a lot of progress has been made over the last 2 decades, and it's impossible to list all the different contributions, but there's been advances in the ability to calibrate these devices. There's been advances in doing rapid calibration, some of the work that Dr. Brandman has done. Some of the work that we were able to do in animals is show that with the advent of neural networks, we're actually able to decode the signals from the brain a little bit better. And when you compare some of the decoded signals with nonlinear algorithms, they tend to perform better than the signals that are decoded from the brain with just linear algorithms. And so this is showing an animal with microelectrode arrays and motor cortex able to control fingers with higher velocities and acquire targets more quickly than the same animal with the simpler decoder type. And this is just one example. There's countless groups that have used these neural networks to improve the ability of their brain-computer interfaces. There's also been work in making these devices robust to instabilities. There's been advanced computational models, and then there's been advances in surgical techniques, and there's added modalities for brain-computer interfaces as well. And when taken all together, it kind of leads us into some of the modern era of brain-computer interfaces. And the 3 applications that I'll highlight are the control of robotic arms, the control of digital interfaces, and then speech neuroprostheses. So now I'm showing you a demonstration of, this is the same group at Pittsburgh. Instead of the user with paralysis just controlling a robotic arm with vision-only feedback, now the user actually has arrays implanted in somatosensory cortex, and when the robotic arm engages with the object, it engages these mechanoreceptors, which send a signal back to the computer, and the computer then injects current through the microelectrode arrays in somatosensory cortex, which the user feels as percepts or sensations in the hand. And the point of this study is to show that when you have this feeling of touch, you're able to manipulate objects a little bit better. And so the video on the left is showing that the objects are being passed more quickly than the video on the right. Some of the work that we've done, when I was at Stanford as part of the BrainGate 2 clinical trial, is looking at decoding individual finger movements, continuous decoding of these finger movements, but then when coupled together with the robotic arm could restore not just single effector movements, but also fine motor movements that are important for a lot of the things that we do as humans, as far as typing, even suturing, things that we do as surgeons, playing instruments. So trying to add this capability as well. Here's a demonstration of a brain-computer interface controlling a computer. And so this is the video on the left is a participant at Stanford controlling a computer cursor. You can see that the control is now much better than some of the early videos. And the important thing is this is real-time control. This is low latency control. This is allowing the user to change and adjust their trajectories in real time based on the feedback that they get. And the video on the right is work that we did with a human participant with paralysis controlling a quadcopter. I think that in addition to allowing for 2D cursors to move on the screen, if we really want to restore the function to that of people that don't have disabilities, they're interacting with computers with multiple different fingers, with typing, with bimanual video game controllers. And so our goal here was to enable the control of multiple different finger movements, not just a single 2D cursor. And so this person is controlling a virtual quadcopter and flying through a spontaneous obstacle course with low latency, probably about around 100 milliseconds or less from when the person thinks to make a movement to when they can actually actuate the movement on the screen. The other application I'll highlight is speech neuroprostheses. I will just kind of, obviously Dr. Channing has really done pioneering work in this field, and I look forward to hearing his comments on this area. But recently we've seen a lot of progress in this area using brain-computer interfaces between multiple groups across the country. I'm going to highlight some of the work that was done at Stanford and UC Davis. So you're looking at two people with ALS and severe dysarthria. So they know what they want to say, but they're not able to control, the voice and be able to produce the sounds of speech. And so they have brain-computer interfaces implanted in precentral gyrus and face-preferring areas. And when they attempt to produce speech, the system is able to kind of classify what types of movements they're making and then construct it into sentences. And so the sentence is displayed at the top of the screen, and the user here, for example, is trying to replicate that sentence, and it's being displayed at the bottom of the screen. And some of the recent results that Dr. Brandman and his group at UC Davis have shown is that it functions quite well as an assistive device, functioning around 99% accuracy at about 50 words per minute. Okay, so sorry, just a little zoom snafu here. So kind of future work, the goal, ultimately for brain-computer interfaces is I hope in 10 years, we're not just talking about 2D cursor control. I hope we're talking about restoring able-bodied function for people with disabilities. I hope that we're talking about whole body restoration, not just, individual cursors. I hope that we also have options to treat novel indications. So not just spinal cord injury and ALS, but looking at other diseases like stroke, cerebral palsy, and other things that brain-computer interfaces could potentially be a therapy for. There's obviously technical challenges as well. We want to develop devices that are very practical, that people can turn on and they can just work, and they don't need to be recalibrated. But specifically, we want to do this in a way that doesn't sacrifice, high-level performance. And I think we're making a lot of progress in this area, and certainly I expect more work to be done. And then I also expect that we'll have a lot better devices. I'm hoping that we'll have fully implantable devices. Why is this important? Because that's what users want. They want devices that they're not tethered to a computer. We want more electrodes and less tissue response, so we have higher electrode yields. This is important so that we can get more signal, so that we can do higher and higher performing tasks. And then we want devices with longer longevity, with less mechanical failure. I was very excited to be part of a study at University of Michigan where we implanted the first Connexus cortical module. It's made by the company Paradromics. This is similar in some ways to the Utah array, except there's 421 electrodes instead of the typical 96. The diameter of these electrodes is also much smaller, 40 microns instead of somewhere around 80 microns for the Utah array. And it's fully implantable. And we were able to deploy it. It actually sits at the bottom of this inserter here. And we can position it with the micromanipulator in the location that you'd like to place it, and then twist the knob, and it deploys into the brain there. And so I'll talk a little bit about the next slide about the kind of the early feasibility study, but something that we're very excited to be a part of. And then in addition to all the other things, we're hoping to continue to innovate on the technology. As I've mentioned before, having small diameter probes is hypothesized to provoke less tissue response. So now what if we take the probe even smaller? So an engineer at University of Michigan, Cindy Chestek, has carbon fiber microelectrode arrays where the diameter is actually 8 microns. And so I'm going to show you a video of this on 256 speed being inserted 100 times into cortex. And this is some work done by one of our residents, Jordan Lamb. And you can see when you look at the cortex after these insertions, it's hard on kind of the naked eye to see where the electrodes are even inserted. This is just a slide from earlier work 5 years prior that shows that kind of chronically implanted array using these types of probes do have a very reasonable or reduced tissue response as well. So, a lot of things that we're doing as a field to try and drive the things forward. I'll talk just real quick. I know I'm really short on time, 30 seconds or so, about 2 clinical trials that we're doing at the University of Michigan. One is kind of your typical study looking at people with motor and speech disabilities using the BlackRock multi-port neuroport system, trying to advance this technology, demonstrate safety, and advance brain-computer interfaces. But then more recently, this is a multi-site early feasibility study that Dr. Brandman mentioned for people with communication disorders. So using the Paradromics cortical module connects this system to a plant and speech areas of cortex and decode someone's ability to communicate and display that on a screen. The exciting thing is very recently we actually started recruiting, or started our phase of recruitment for this clinical trial, and we're very excited to see where this goes. So I won't waste too much time talking about conclusions, but obviously these guys have come a long way, and there's a lot more work to be done.
Matt, thank you so much for that. That was terrific. I think we'll move right over to our next speaker here. So Ben, if you'd like to get started.
Terrific. Thanks so much for having me. Are you seeing my slides okay?
Yes, we see your slides now.
Terrific. Well, it's great to be here with this group, with colleagues who've done so much to advance the field. It's, as a few people have already said, a very exciting time for the field of brain-computer interfaces, and definitely makes sense for the functional neurosurgery community and the ASSFN to be at the leading edge. At Precision Neuroscience, we're focused on building a scalable infrastructure for brain-computer interfaces. And this means interfaces that maximize information density while minimizing biological cost, and that are designed not just for performance, but for deployment at clinical scale. Really, we're trying to advance the standard of care in brain-computer interfaces. And it's a very collaborative endeavor, of course. Today, I'll be sharing just a little bit about our viewpoint and recent progress and our experience, as David suggested, working with the clinical community and with industry incumbents to try to develop a scalable path forward for brain-computer interfaces as a clinical standard of care. Just a word, my disclosures, as David mentioned, I'm assistant professor of neurosurgery in the neurosurgery department at Mount Sinai in New York City, and I'm co-founder and chief science officer at Precision Neuroscience. And I'd also like to just mention that we received a 510(k) clearance last year for a percutaneous version of the Precision system, and I'll be showing some results from clinical studies involving this system, but all the work presented here was performed in an investigational capacity under local IRB approvals. And just to thank you in advance, because in these short talks, there's a tendency to run out of time. So I just want to take a moment to thank our participating sites, our patients, the Precision team behind this work that I'll be presenting, and to the ASSFN for the invitation to speak. Brain-computer interfaces represent an evolution of a paradigm that's familiar to the functional neurosurgery community already for several decades. But in neuromodulation today, for the most part, stimulation equals therapy. In brain-computer interfaces, for motor prosthetics, for communication restoration, and for other applications, neural decoding, or inferring intention from the electrophysiology, is the therapy. And neural decoding is no longer a laboratory science technique, and Matt did a beautiful job of showing several decades worth of work in the field where the the brain-computer interfaces that we know really emerged from decades of deep research into electrophysiology at the individual neuron level. But neural decoding is no longer a laboratory science technique, and now it can be performed in the operating room in real time. And at Precision, we've deliberately iterated towards an intraoperative workflow that reflects standard practices from awake deep brain stimulation and awake cortical mapping, as these techniques are practiced today in trying to devise a workflow for brain-computer interfaces, over the next couple of years. And I'll show some examples as we move forward. But the central question now is no longer, can we decode? The real question, especially for this audience, is what system architecture will scale safely to tens of thousands of patients and beyond? And Matt kind of set that up, and I'd like to present some thoughts on that question. So neural decoding requires multi-electrode array data, and each available array architecture makes trade-offs across resolution, coverage, biological cost, and other factors. And at Precision, we have focused on developing an architecture that maximizes information density while minimizing tissue disruption. And if you think about what determines adoption of a new technology in practice, of course, safety and efficacy, but beyond that, I think it's a combination of reproducibility, biological tolerance, for us, functional area coverage, risk profile, regulatory considerations, and all the practical aspects of manufacturing a very high-tech new technology. And of course, in neurosurgery, we all know that to a certain degree, elegance and scalability somehow determine what survives. And at Precision, we believe that procedures that are biologically tolerated, surgically reproducible, and manufacturable at scale become the standards of care. So this is an illustration of the Precision system. And our fully implanted system, which you can see here, it looks very similar in many ways to standard neuromodulation systems of today. It comprises a high-resolution, thin-film cortical surface array. That's about 20 microns in thickness, very conformable to the cortical surface, and contains 1,024 platinum microelectrodes. There's a hermetically sealed skull-mounted signal processing unit, a tunneled lead, just like in DBS, and a chest wall unit that contains a rechargeable battery and antennas for power and data telemetry. And using a high-resolution electrode array of the form that I just showed has certain advantages from a signal processing standpoint, and I just want to kind of touch on them here. And what you're seeing here is on the left a color map of surface voltage in time, and each color dot represents one electrode. The spacing here is 400 microns, similar to what you would get from a Utah electrode array. And we can compute features in real time, in time and frequency domains, and also in two-dimensional space. And some of these features are quite uniform across patients due to the uniformity of the array geometry from patient to patient, we're learning. So treating the electrophysiologic data essentially as a high-speed video enables us to employ acceleration techniques from modern machine learning, which were really built for analysis of video and images. So I'll show you just for a minute, as I mentioned before, a glimpse of the intraoperative workflow that we've been evolving forward with a number of our clinical collaborators here. You're seeing the precision electrode array being implanted as part of one of our clinical studies in a awake DBS case. This patient had an STN DBS for Parkinson's disease. It was done awake, and in these contexts, it allows intraoperatively for the chance to train and test a decoder. So in a patient like this who actually has the ability to move and give us gold standard ground truth data for their ability to move and intend or imagine movement, this has enabled us to really learn how to, in the intraoperative setting, train models and implement them on a scale of minutes on small amounts of data, and to do it with reasonable accuracy. And in this case, and I'm foreshadowing publications that will be coming out hopefully over the next couple of months by a number of our collaborators, we see an example of a consistent finding, which we feel was important, in that movement and movement intention are decodable from the cortical surface using high-resolution microelectrode arrays. Of course, as more data is added over days, hours, and days, the performance improves, but the basic elements of two-dimensional motor control are available within minutes. And this is very important because within the intraoperative workflow, electrode placement can be verified anatomically, just like it is in DBS. Initial training data can be acquired, and likewise, within minutes, the awake patient can acquire basic neuromotor control. So if you think about moving this forward into a kind of workflow for patients with paralysis undergoing placement of a device for motor prosthetic, this kind of intraoperative confirmation, we believe, will be critical to early neuroprosthetic workflows, especially during the clinical trials and early years of experience to ensure reproducible outcomes. Also on the road to clinical scale is really a very strong emphasis on manufacturing and manufacturability and quality control. The supply chain in the medical device industry for these high-resolution electrode arrays and other associated components really doesn't exist. So behind the scenes, there's a lot of work that goes into ensuring that this can be done robustly, consistently, at scale. And as I mentioned, last year we achieved a 510(k) clearance for the precision electrode array that you're seeing here, and that has facilitated collaborations with a number of investigators, including some in the audience today across the country. And it's a privilege working with you all, and I really thank you for the collaboration. We now also have a partnership with Medtronic, initially focusing on bringing high-resolution electrophysiology to the Stealth platform and bringing Stealth guidance into the targeting workflow for brain-computer interfaces. All of these really with the intention of standardizing and delivering consistent results in a way that's in keeping with workflows that we already know from functional neurosurgery. And as David mentioned at the beginning, I'd like to just mention here the work of the Implantable Brain-Computer Interfaces Collaborative Community, a great group with an unfortunate kind of initialism name. But this is a group that last year drafted an initial set of sort of proto guidelines for what might emerge over time iteratively as kind of clinical practice guidelines for the field of brain-computer interfaces. And this year, the group is focusing on thinking about what would be required to stand up BCI centers of excellence. And I would really encourage and implore those of you with interest and expertise to contribute some of your time and knowledge to this evolving discussion. And hopefully that will help shape the standards of care in a productive and responsible way. Importantly, following the 510(k) clearance, we have been deploying investigational percutaneous versions of the precision system in pilot clinical studies at multiple centers across the country. And we're also seeing cross-site collaboration. And this type of real-world data is very exciting to see. It's invaluable, and it helps sanity check the work when you have reproducible results across multiple surgeons, multiple sites, multiple clinical teams, and multiple deployed systems in the field. Also, this kind of work kind of encourages exploration, and clinical studies with the Precision platform have investigated surface electrophysiology for hand, sensory and motor function, speech and language related cortical function. One of the nice features of these thin film arrays is that they can be deployed intracellularly as well. And they have been deployed on brainstem and spinal cord surfaces. Real-time processing of high-resolution electrophysiology, neural decoding in the context of a motor or speech prosthetic is finding applications now across a number of areas, not all of which we would have anticipated from the motor and communication prosthetics use cases. And this too is really exciting. And personally, and the whole Precision team is very excited for collaborating sites to continue sharing their findings publicly in the months ahead. One last thing I'll just make note of kind of along these lines is that when we have multi-site collaboration with really uniform electrode arrays being deployed across different surfaces within the central nervous system, this allows for population scale mapping. And we now have the ability to combine multi-patient data sets and large area sampling and to use these for understanding patient-to-patient variability at scale that was not really previously possible. It was not really, we have these standard atlases that we use in functional neurosurgery, but we don't really have standard electrophysiologic atlases at this kind of scale. So it's exciting to see this emerge both for standardization of clinical care and also for discovery. I think there's a lot to say. There's so much to say on probably each of these slides, but We're only allotted 15 minutes this evening. So as a final thought, I'll just say that for many of us in the field who've been involved for, a couple of decades, there's a clear shift in the locus of the conversation. And I just want to point it out that this type of presentation used to take place in the academic societies, the SFN, for example, and being here at ASSFN is a reflection of progress in the field as a whole. There's certainly more potential for discovery than ever as the platforms advance and mature, but importantly, we're having this discussion in this forum because brain-computer interfaces are becoming a clinical reality and hopefully before too long, a standard of care. So I want to thank you all for being here this evening and to thank the ASSFN for inviting me to present. And I'm looking forward to the discussion after Eddie's talk.
And thank you so much for that talk. I really enjoyed that. Let's transition to our third speaker now, Eddie Chang from UCSF.
Hi, everyone. Good night or good evening. I wanted to start by just thanking Dr. Ben-Haim and Dr. Brandman for organizing. This really is a timely topic, and it's a really exciting time. I think we're really at an inflection point for thinking about how to translate a lot of the functional neurosurgery and physiology into brain-computer interfaces. And David asked me to really focus on, the work that's been in my lab. And as a disclosure, a lot of the work that we have done in lab is the basis of a company that's still in very early stage that I co-founded called Echo Neurotechnologies. The content of this presentation actually is not about the company at all or its technology. It's really what we've been doing in lab towards understanding the basic science of speech production and language in the human brain. And this has been a really interesting journey for me over the last 15 years. When I started my career here at UCSF, I was definitely interested in BCI, but a lot of it was focused on cursor control. And Matt showed some really cool historical vignettes about sort of this steady progression in terms of cursor control, and ultimately its use for other applications like writing and texting, etc. I've been really interested in speech my entire career, and one of the particular reasons why speech as a BCI I think has taken some time is that for many, many, many years, people just thought it was too complex, like a really more of a cognitive kind of phenomenon. But we took a different approach towards this over the last 15 years. We've really focused on trying to understand the speech motor control and really thinking about speech as a motor problem and breaking it down. And that level of that approach has actually helped us to really understand sort of what are the building blocks that have allowed us to understand the neural code for things like consonants and vowels. I'm not going to go over this work, but there's, like I said, a decade and a half of basic science using high density electrocardiography in the epilepsy monitoring unit over the last 15 years, really just to understand what's the nature of cortical activity that I think links the control of our lips, jaw, tongue, larynx to this coordinated control of our vocal tract that gives rise to consonants and vowels, which by themselves have no meaning, but in combination give rise to all syllables, words, and sentences. And so you can think of speech as a motor system that allows us to express essentially infinite meaning. One of the early papers that we published actually on the code for consonants and vowels and the neural activity related to the vocal tract led to a lot of surprises scientifically. But one of the early questions was really about whether or not you could actually use this kind of electrical code, this dictionary that we discovered about 10 years ago, to essentially translate that neural activity into words for someone who is paralyzed and unable to speak, like physicist Stephen Hawking. And I think at the time, I remember having really distinct conversations with some of my friends in the BCI world, like Jose Carmena, who was a collaborator, and also even Christian Shenoy, who was a close friend. And it just seemed like, something that was just so impossible to think about because we're still at the level of cursor control. And why I'm saying this now is because what was once seemed impossible in science fiction actually has been done now, not just by our group, but many other groups, validating these results and at very, very, very high levels. So it's very exciting for me to see something even in my career that has gone from concept to demonstration. One of the first demonstrations was really this work that we did in the epilepsy monitoring. And the reason I'm bringing this up now is because a lot of this was actually created in the EMU in our functional epilepsy practice using high density arrays. And in my practice, I still use a combination of stereo EEG and grids, really depending on the case. But this was really one of the first demonstrations of using brain activity to synthesize words that you can hear. And it's no coincidence that this happened, about 6 years ago, 7 years ago. Um, at this time, it was not really about the neural activity. I think it was nice to have high density recordings. These are 4 mm spaced electrodes, but we've had that, as many of you know, for decades. I think the thing that's really changed is really um, the very, very rapid progress in machine learning methods, in particular recurrent neural networks for this particular project. And so what we did in this project was essentially use a machine learning method called an LSTM to translate neural activity into a virtual simulation of vocal tract. And then from that simulation, we were able to translate that into sounds that you can hear. And so what you're about to hear is essentially, that first demonstration. It was published again 7 years ago. It was, I think, just some of these examples were really above the threshold of intelligibility. But I think what was exciting about this to us was that you could actually just use some of these standard epilepsy arrays that we were using in the hospital and with machine learning methods and actually get stuff that's starting to sound intelligible. So you're going to see the activity on the brain when someone is speaking. Again, this is an epilepsy patient, not someone who's paralyzed. And then you can see it driving essentially, the virtual simulations of a vocal tract. So we're using an articulatory model of speech production, essentially something that we've been working on for 15 years, understanding how that code works. And then that model is generating sounds that you can hear. You're going to first hear what is synthesized by the computer and then basically what the person actually said.
The proof that you are seeking is not available in books.
The proof that you are seeking is not available in books.
The proof that you are seeking is not available in books. Shipbuilding within my fascinating process.
Shipbuilding is a most fascinating process.
Shipbuilding within my fascinating process.
So one of the things that I wanted to just point out here is that for the neurosurgeons who do brain mapping, it's probably very obvious to you that we're recording from the ventral, precentral, Rolandic areas. So these are the areas that correspond to the mouth, the lips, the tongue, the larynx, etc. And so what you are really seeing is not necessarily like a decoding of someone's thoughts. It's not even a decoding of the representation of words, but it's really more of a representation of the body map that's being played out. So that's really like a dynamic representation of part of the homunculus that that gives rise to our ability to produce words. So this was very exciting to us. And like I said, it was the first time that we or anyone else had been able to really synthesize speech in an intelligible way for some of these examples using electrocorticography. And this was in the epilepsy monitoring unit. So we took those results and sort of that insight, and I would say it's the insight that was really more important to apply for an FDA trial, approved trial called the Bravo trial. This was just as part of an experimental IDE. for about 6 patients at UCSF. And it's called BRAVO. It stands for BCI Restoration of Arm and Voice. The basic idea was to essentially evaluate using ECOG approach, using implanted subdural grid electrodes, first 128, now 253. And we attached it to the same percutaneous port that the Utah array is normally connected to. So we worked with the device manufacturers. We took PMT epilepsy grid and had it attached to the BlackRock percutaneous port. We also had approval for the Utah array to do that, but really one of the things that we wanted to do was really focus on something that was clinically really like more clinically viable approach using existing technology. So we ended up proceeding with the ECOG approach for this. Something that's also kind of unique about this trial was the first chronic ECOG BCI for beyond a 30-day. So we really just didn't know what this would look like for weeks and months. The basic idea is that the array would cover the entire lateral motor cortex, covering both arm and speech areas, both motor and sensory, for people who have severe paralysis. And Matt alluded to something kind of earlier about, whole body, and that's something we're very interested in, which is for many of these folks who've got severe ALS and brainstem stroke, you actually can benefit a lot from, multimodal whole body representation, and that's something that we're thinking quite a bit about. So the first paper that we published from this using 128 channel array was published in New England Journal in 21, and it was the first successful decoding of speech in someone who had a severe form of anarthria from a brainstem stroke. His name is Pancho, and he was in the study for 6 years, which was the limit that we were allowed through the FDA. But basically, the way he communicates is using a device that's called an AAC. It's an augmentative assistive communication device. And so many of our patients use this after devastating brain injuries. It can produce for him about 3 to 5 words per minute. He communicates by moving the stick across the screen. And one of the main complaints that I'll get to in a second is that this allows people to create text, but there's no emotional expression. There's no expressivity. And we've done a survey of many of our patients, and that's something that it's not just about the words, but it's also about the expression. This is Pancho's sagittal MRI, and this is just to show you the really severe encephalomalacia and his brainstem and the pons in particular that he was left with after a vertebral dissection. So this is, again, fairly primitive what we did, I would say, because what we took was off-the-shelf epilepsy grid. We attached it to the BlackRock array. It was the first chronic ECOG study for BCI using these high-density kind of surface arrays. And this is just a illustration of the signals that are coming from Pancho's brain to a computer where it analyzes his intended speech and then uses a language model to help put it all together and puts it on a screen. If you look at the parts of the brain that were most important for decoders, they come from these two particular areas. In the middle precentral gyrus, this is an area that we've identified is really important for speech motor control. And it's right underneath the hand area, and then an additional area that is closer to the ventral central sulcus, which is, I think, encoding more articulatory information. Okay, so many of you have seen this video, but this was essentially one of the first demos that we had. Someone is writing to Pancho in the lab. The signals are coming through this HDMI, and the blinking dots means that it's detected that he's trying to say something, and then the rest of the time is just inference trying to decode what particular word. And again, this uses a language model to accelerate and correct incorrect words in the context of a sentence. This was about 15 words per minute, and it was faster than what he already had. It was a limited vocabulary of 50 words, but I I think this was a really important milestone for the field because this was essentially the first successful speech neuroprosthesis application in someone who was paralyzed and was really just the start. And things have accelerated tremendously over the last 5 years. One of the reasons that we went with the ECOG was to evaluate its long-term stability. These are high gamma signals from a single electrode. Ben talked about the virtues of, essentially doing surface recordings and the stability of that. And so, these are just high gamma, high frequency responses from one electrode, as Pancho is trying to say the word goodbye, that are stable over weeks, months, and we now know years. Another participant was Anne. We used a 253 channel array and got, really a step function improvement. So the kind of things that Ben was talking about, that higher density, more channels does mean more information. It does mean better performance. And these are things that we're looking at very carefully, like what is the optimal amount. In this calibration test, where we have people essentially trying to say the NATO code words like alpha, beta, Charlie, etc. It's just a standard set of words that we can quantify. We could get up to about 90 to 100% accuracy in about 10 days of training. And then on top of that, we could essentially fix the decoder at 15 days, I guess, after we started training the algorithms and just keep it fixed. So it's a very stable signal. I'm not saying it's like 100% stable. Over some months, you sometimes do have to recalibrate things and things get better, and there's some shift in plasticity. I don't think it's because of movement of electrodes, but we've discovered that the underlying representations in the brain actually can shift over some time, which is a really interesting thing when you've got arrays implanted for that time. So what we did with this participant, Anne, who had a very similar situation, she had a brainstem stroke. We essentially built three simultaneous AIs. One was using brain to text, one was brain to voice that you could hear, and then we also built a virtual avatar. And the reason we did that was for two reasons. One is that we wanted to give people feedback like sensory motor, or like a visual motor articulatory feedback, because that's essentially how we are used to speaking. We're not used to having text as our feedback. We really are thinking about it as an articulatory movement, and we're currently using that our articulatory visualization through an avatar to give people closed loop feedback on how to improve their speech BCI. And so here's some of the early results. On average, we were able to get about 78 words per minute. In some trials, it's much, much faster than like 100 words per minute. Here, the blinking dots is not single words. It's actually like a detection that when she's trying to speak, and then it's essentially decoding the whole sentence. And in this particular study, about 6 years, like that we published in 23, the algorithm essentially looks at the entire sentence and then the neural activity for a full sentence, and then applies a language model and then displays that. And so there's a lot of processing that's decoding every individual phonetic element and then puts that together using a model, and then uses the statistics of English to aggregate that, correct it, and make it more accurate. So this is one of the demonstrations of high performance speech decoding that Matt cited earlier. And thanks, Matt, for the shout out on that. And things have continued to improve with work from David and Sergey with, a different subject. There's so much to be done. This is an example of the avatar.
Be a few minutes.
So again, the reason we did this was to give people real-time feedback. So like this is the way a speech therapist works when they're working with patients that have lost ability to speak. They like use feedback tools. They think you have patients think about the control of different movements, and that turns out to be something very important. Something I alluded to earlier is that the loss of emotional expressivity for the vast majority of patients who have severe paralysis chronically is not just the inability to communicate through words, but it's also, interestingly, one of their top things is emotional expressivity. because AAC devices cannot really allow you to express a lot of emotion. And so, in addition to showing that we could translate those ventral uh, facial movements into speech, we could also use them to decode non-facial gestures and facial expressions, that kind of thing. This has now become much faster. This is a paper we published last year where the decoding is being done very low latency streaming. So this is basically like,
Before midnight. Let me tell you what I did.
so this uses a different architecture called an RNN transducer model and allows us to essentially try to do the inference in real time, not waiting for the full sentence, but actually sort of doing a lot of the decoding in real time. And this is modeled after Anne's voice. We've also shown that you can use these kind of brain activity patterns to decode whether someone's trying to speak in Spanish or in English. We basically have built a bilingual speech prosthetic. And so just to summarize, in this UCSF very small trial, but we've learned a lot from this using this adapted ECOG device. Um, a lot of different examples from brain to text, brain to avatar and voice, brain to facial expressions, brain to text. And some of the projects led by my really close collaborator, Karnish Ganguly, who's a neurologist who focuses on BCI. He's working on a different part of the array that has to do with arm and hand. And so he's had some really cool demonstrations using Pancho and Anne controlling robotic arms. So a lot of this can be done just through one array, but covering multiple areas. So last thing I'm just going to conclude with, and I think some of the other speakers have also concluded on something similar, which is as we think about clinical translation, there's definitely things that we want in terms of the performance. That's what most people are focused on right now in the academic sphere, which is like how good can this get? But the more important questions, I think, long term are going to be about the robustness of the technology. As functional neurosurgeons, we really do expect this to work um, across all of our patients and with high performance. We want it to be easy and fast. You can't have essentially a research team supporting all of this. It has to be really something that can be used autonomously and across a lot of different indications. And then from a surgical perspective, we want it to be, of course, fully implantable, not this kind of percutaneous thing that I showed you, but a fully wireless one. And that's something that we're working towards. This last point I think is absolutely critical is that these technologies are safe and easy to implant and explant and that are stable over time. I think that this is going to be very, very important, especially for patients that already have weakness. You don't want to risk making them impaired, like more impaired. Some patients are partially paralyzed, not like completely paralyzed. And if you injure the part of the brain that you're actually trying to read out, that can be a problem. And so this is actually not trivial, sort of like how to achieve all of these goals. But I think that these are just some of the things that we think should be optimized, and I put this in this perspective. So I just want to thank everyone for being here and opportunity to just share in this conversation.
Perfect. Thank you, Matt. Thank you, Eddie, and thank you, Ben, for speaking today. That was phenomenal. I've seen each of you speak before, and today was absolutely terrific. I wanted to open things up for questions now. I have about 15 minutes of questions. For logistics perspective, for the audience, please type your questions into the chat. I'll do my best to read out the questions and then assign them accordingly to the speaker. We may not be able to get through everyone's questions, so apologize about that ahead of time, but we'll do our best to kind of go through that. As people prepare the questions in the chat, I just want to make a quick comment about where we are in the field and where things are going. As you've heard today, we've heard about a few different technologies. Matt referenced his first in human translation for the paradromic system. Eddie's talked about his technology with ECOG moving towards his company Echo. Benjamin's talking about his company, Precision Neurotechnology. There's, of course, Synchron, which has completed early feasibility studies looking at the safety profile of their chronically implanted system and their stentrode. And of course, Neuralink, which has looked at their device, which has multiple implanted threads able to record from multiple cortical layers simultaneously. And so one question you may be asking is, what's the right technology to use? And the answer ought to be, well, it depends. I think we're going to come to a point very shortly where hopefully as we as surgeons are going to have multiple devices we're going to be able to prescribe for patients depending on what their indication are. Looking at my own reference and experience with epilepsy, when we meet patients with epilepsy, sometimes we prescribe a VNS or a DBS or an RNS, we'll prescribe a cortical resection or laser ablation. And I think in the future, I hope, we'll be to a point where we can prescribe multiple different kinds of devices depending on what someone's looking at. Perhaps someone's not interested in a craniotomy. Well, in that case, there could be device A. Perhaps someone's interested in speech restoration. They go for device B or C depending on the circumstance. And so it's actually a very exciting time because there's a lot of companies that are developing fully implanted wireless systems to restore function in people with paralysis. And we're just learning as these technologies are developed, who are the right people to help and how can we help them most effectively. And so with that in mind, I'm going to start by reading out some of the questions. How critical is cortical location for the ability to process neural activity for control of a cursor or prosthesis? I have thoughts about this, but I'd like to pass this off to Matt and Eddie to talk about, does location really matter? So Matt, why don't we start with you?
Yeah, no, it's a good question. I think at this point there's been multiple studies out now where, I mean, I think we know like the general somatotopy of the brain, there's arm preferring areas and face preferring areas. And I think we've seen, several studies, including David and Sergey's study with people being implanted in V6V, which Dr. Cheng talked about 55B as being kind of like the ideal places for maybe putting a cortical array for speech, for example. And then maybe areas of precentral gyrus and hand knob being kind of the ideal places for placing an array for controlling of a cursor or like a robotic arm. But I think there's also a lot of studies out there showing that these areas you can infer information about other parts of the body, even if they're preferred for one type or the other type. There's been studies out at Stanford showing that hand knob does have representation of whole body movements. So I think the answer is as David said, it depends kind of what your application is. If like for example, if and I'll let Dr. Cheng comment on the thoughts on looking at it from an ECOG perspective, but if you're looking at it from a microelectrode array perspective and you have a device that's implanted and functioning as a speech neuroprosthesis, you can also probably decode, and I think Sergey and David have showed, decode cursor control movements from this, and vice versa. So.
Just to follow Dr. Wilsey, I think one of the most amazing discoveries over the last, 10 years with the microelectrode work is that is this concept that that there are these multiplexed, multiple representations for different parts of the whole body across the homunculus. Now, I don't think that that means that's how the brain works or it's causally, the mechanism of how things work. But for decoding, I think it's perfectly valid as an approach. And I think even our understanding of this like delineation of arm versus speech, we haven't seen much speech in the hand area and the level of ECOG, but we definitely see a lot of motor control, in the ventral premotor areas. And so I think the bottom line is the more and more we look at this data, we appreciate that the precentral gyrus is not just the motor cortex, it's really the premotor cortex. And there's a lot more higher order planning that we're tapping into. But I think, location is very important. With these ECOG arrays, we have the luxury of just covering a lot of territory very safely with no, physical risk from that. So whether you're essentially covering, many, many square centimeters or just one, it's essentially the same risk, basically. And I think that's a huge advantage. One of the things that we're starting to look into is actually the sensory representations during these movements as well. So it's not something many people have studied a lot, but in fact, there's a lot of motor-related activity in the postcentral gyrus, actually. And there's actually evidence of sensory responses in the pre-central gyrus. So this is a lot of real estate that I think we're just starting to understand. And location, I think, really does matter.
Yeah, I would just add to that as we get more and more confident in the kind of next generation platforms and methods, we kind of change our definitions for what constitutes the 21st century homunculus. So, we kind of used to defining, somatotopic representation based on a 20th century notion of event-related potentials and cortical stimulation, for example. And now we have a new set of methods, as Eddie was alluding to, where, especially in the context of ECOG and micro ECOG, you can, with essentially zero risk, map those areas, not just for event-related potentials, but also for decodability. And you can ask the question, can you decode, for example, motor intention from area X? And then you can answer that question with, very, very low risk. And I think that's actually going to be a very important part of how the clinical workflow for brain-computer interfaces works moving forward. Just like, especially in the early days, but even, until today, awake intraoperative deep brain stimulation, awake intraoperative mapping for deep brain stimulation, awake intraoperative mapping for lesionectomies. How we do the mapping is going to be a critical part of effective deployment of this technology in the future.
I want to give concluding remarks just about this, and the issue of safety has come up in a few different speakers as part of this. I want to remind the learners, we have some residents and some fellows that are joining in today's conversation today. When we structure clinical trials, we are asking the question, is a device safe? Right? And so one of the things we're trying to learn with these various clinical trials that are recruiting people isn't whether or not these systems are effective, but rather, are they safe for people living with paralysis? And certainly there's ongoing clinical trials that are trying to ask that question. And it's important to recognize that the question of safety is paramount. It's really the sine qua non in moving forward with this technology. But as we learn about the effect of this technology through these clinical trials, we're focusing on really answering that safety question first. And certainly speaking my own experience as part of the BrainGate clinical trial, this clinical trial has been going on for over 20 years. And we've published interim analysis of the BrainGate clinical trial of a implanted Utah array in people with paralysis that's been fairly favorable to that end. So safety is very important, and we will learn that as a surgical community exactly how safe these devices are with these early feasibility studies that are put on with these various industry partners as part of this. So I want to talk now about one of the questions that's come up in the question period, talking about wider indications. And Ben, I want to turn the question to you first. One of the things that we focused on are people with paralysis, right? Looking at people with brainstem stroke, looking at people with ALS. But of course, this represents a fairly small chunk of the population of the people that when you think about people living with neurological disease in general. Ben, what do you think about the next generation of technologies, and who are the people we ought to be helping? And what are the kinds of indications we should be thinking about? And more broadly, where ought we be going in the brain to solve those kinds of problems?
Yeah, I think I appreciate the question, and both Dr. Wilsley and Dr. Chang, mentioned their thoughts on this in the concluding slides. I think that this is a question for the community as a whole, and I think it's important that we be developing technologies that are safe and can be distributed to and used by a community of experts that have insight into specific disease conditions and ideas about how to use the evolving technology. I don't think it's necessarily the role of any one individual or team or company to prescribe the roadmap. Certainly, I have my ideas, and Precision as a company has our ideas for what makes sense to do next. We have to. But I think, we have to be humble enough to know that there's going to be great ideas about what to do with the technology as it evolves. And then, Eddie mentioned in his retrospective that people used to think that speech was impossible, and then he had an insight into how to crack it from a motoric standpoint, and all of a sudden it became much easier. So I think that, when there's a platform and techniques available, people have insights and ideas of how to use them that are not always apparent, when the technology is developed. And I'm positive that that's the case for brain-computer interfaces, and I'll say from our work across, many sites now, we're already seeing that. We're already finding unexpected things. We're seeing people with ideas that they want to pursue. And I have to say that's very exciting. But to answer your question directly, I'm very excited about stroke as a kind of a next step uh, beyond sort of ALS, brainstem stroke, and spinal cord injury. I think that the kinds of partial paralysis that we find in more common forms of MCA territory stroke, for example, some of them will be amenable to addressing using brain-computer interface technology. I think in particular, ECOG and micro-ECOG, which have the ability uh, to map areas of residual functional cortex, and perhaps even to stimulate areas that that may be rehabilitatable. I think that's an open question. So to me, that's a much more prevalent disease state, and it's one that has seen tremendous change in our lifetimes. So I think there's both a technological sort of push and a clinical pull to innovate in stroke and stroke rehabilitation, and I think that's an area where we'll see some work over the next few years.
Matt, I want to turn the next question over to you. A lot of what we've heard today from Ben, Eddie, and yourself has been about reading out from the brain. In terms of your historical conversation about BCI over the last 20, 30 years and beyond, can you talk a little about writing into the brain? Where are we with that kind of technology? And what are the obvious use cases where we can actually use a closed-loop BCI system to restore sensations to people?
Yeah, that's a good question. So, most of the BCIs that have looked at recording information out of the brain for trying to infer like movements or speech have, especially for movement, have looked at vision-only feedback. And the studies, I think, out of Pittsburgh, for example, being able to write somatosensation back in, I think there's been a lot of progress. Like I think initially it wasn't really clear like how to really focally generate percepts on the hand, for example. I think that there's been a lot of work at looking at kind of limiting the amount of stimulation, looking at ways to make stimulations seem more naturalistic and less artificial by potentially adjusting stimulation patterns or limiting the amplitude. But I think writing into the brain also, we have to think about, and we didn't really talk about it in this talk, but stimulation in occipital cortex or for vision. And I think this is potentially, this has been looked at in the past. This is a potential area of interest as well.
Cool. We'll be wrapping up very shortly. I do want to ask one more question. This one I'll ask towards Eddie, just about the next generation of BCI researchers. So one of the questions in the audience is, how do you get involved in this kind of thing? You're a neurosurgeon, you're a medical student, you're a resident, you're a fellow, and you're thinking you want your career to grow towards BCI technology. What are the kinds of things you can do in training to kind of set yourself up for success in your future career?
That's such a wonderful question, and it's such a wonderful one to end this with, which is about the future. And I think it's very clear that we are at the very beginning of all of this. And one of the things that I really like about this field of BCI is that it covers a lot of different territory, from the science that underlies the brain coding for movement or emotions, cognition, etc., to materials. I think all of us would admit, like very, very, quickly, that some of the material challenge has not been solved. There's a lot of work to build, ultimately a high fidelity, a high bandwidth, sustainable, robust interfaces. And everything has some trade-offs, at the end of the day. So I think that quest is really there for those future material engineers. One thing I alluded to earlier is that even with very primitive approaches. Like we just took one of these PMT arrays for epilepsy and applied it here. It was the first time, and I think it was appropriate because it was like a known commodity and a safe one. And things are much, much better now. But I have to say, like machine learning, AI has been the absolute game changer for all of this. And so you can take relatively weak signals that are very hard to see what's going on. Um, and with enough data, you can do very, very, very powerful inference. So my point about this is that one skill set that you can't go wrong with is really the quantitative computational ones and really understanding, everything that we've used, and I think this is true for many other groups, has relied on machine learning methods that have improved speech recognition, LLMs. All of these things we're actually using to translate brain activity into words and sentences, and they're very, very similar to the algorithms that are being used across ML. So this is just such an exciting time for people who have different interests and skill sets to come into it.
Yeah, that's very well said. Um, I'll also say that some of us on the panel, for example, are fellowship program directors. And if you're an aspiring functional surgeon interested in kind of BCI development, it makes a lot of sense to go train with a neurosurgeon who does BCI for a living as well, as another way to kind of get engaged in this from a clinical perspective, and then use this as an opportunity to understand what your specific interests are and think about incorporating research as part of your additional training to kind of set yourself up for success and kind of building a future career in this. So with that in mind, I'll pass this off to Sharona for closing remarks.
Wow, well, thank you, David. That was really a fantastic session. I just wanted to make a quick announcement about our next ASSFN webinar, which will focus around a new series around epilepsy surgery. That webinar will take place on March 11th, and that first part will be around surgical decision making at the same time as this webinar. I really want to extend a thank you to our outstanding speakers this evening. For a truly fantastic session. Thank you to Dr. Brandman for both moderating and helping to organize this session. And I really want to thank you all for attending this evening. Good night, everybody.